Welcome to the lecture series in food biotechnology. Food biotechnology. This is a novel term for something which has been existing from the time man started cultivation, selective breeding and improvising on the crops for better yield, better nutrition or for some other aspect. In this particular topic, today we are going to talk about one major aspect, single cell protein and its applications for human nutrition. The term single cell, when we start talking about the continuous rise in the global population, to feed that increased amount of population, we need to produce more food. So, on one hand, food production has to increase. At the same time, when population increases, most of the cultivable lands or the agricultural lands get converted either into housing area or they get converted into places where industries are kept. So, you need to balance this. You require increased food production, you require urbanization. At the same time, you need to know what to do in agriculture to produce more food, so as to feed the growing population. The conventional practices in agriculture and animal husbandry have been unable to fulfill the nutritional requirements of the present population. This in turn has resulted in malnutrition owing to the deficiency of protein in food. Humans and animals consume protein as a source of nitrogen and essential amino acids. These essential amino acids are used to build new structural and functional proteins, for example, enzymes and hormones, and these enzymes and hormones are required for them to survive. In extreme conditions, proteins can also be used as a source of energy. The nutritional value of a protein is determined by its amino acid composition. 20 amino acids are commonly found in dietary protein, of which several, for example, phenylalanine, valine, threonine, tryptophan, methionine, leucine, isoleucine, lysine and histidine, with arginine, cysteine, glycine, glutamine, proline and tyrosine. These five are also extremely beneficial, and these cannot be synthesized by humans or animals. Therefore, these are essential and have to be supplied through the diet. So, these essential amino acids, which cannot be synthesized either by the humans or the animals, have to be necessarily supplied by the diet taken by the organism. An increasing demand for meat and dairy protein will require improvements in agriculture production as well as an openness to new sources of protein, both as animal feed and for direct human consumption. Animal and dairy production have been increasingly steadily over recent decades, and theoretically this can continue to do so to meet the expected demand. Plant protein is converted rather inefficiently into meat protein. Approximately 6 kg of plant protein is required to produce 1 kg of meat protein. Hence, increasing meat production to match the growing demand is ultimately not sustainable. The Western world is also interested in developing healthier food with optimal amino acid composition and low but good quality fat combined with ethically sustainable production. These are typically non-animal based, environmentally friendly processes, but these may include novel processes like the production of cultured meat, in which meat protein is more efficiently produced in vitro rather than by growing an entire animal. Protein can also be provided through the cultivation of various microbes and algae, preferably those which contain more than 30% of protein in their biomass and which can produce or which can provide a healthy balance of essential amino acids. So, instead of growing an animal and then finally slaughtering it and using it as a source of meat protein, if you have some methodology whereby the same meat can be synthesized in vitro, or you have some other source which would provide a more balanced amino acid composition which is more nutritive, that would be more sustainable in the case of feeding the population. Microbial protein is generally referred to as single cell protein, normally represented as SCP, although some of the producing microbes such as the filamentous algae or filamentous fungi may be multicellular. So, irrespective of whether it is the filamentous fungi or filamentous algae which are the source, commonly we term them as the single cell protein. This refers to the crude, a refined or an edible protein which is extracted from pure microbial cultures, dead or dried cell biomass. So, the term single cell protein is derived from microbial culture. This culture can be dead or dried, we get the cell biomass, which is, this can be crude or it can be refined, and this is used as a supplement to feed the animal or human population, and this can be used as a protein supplement for both humans and animals. Microorganisms like algae, fungi, yeast and bacteria have very high protein content in their biomass. These microbes can be grown using inexpensive substrates like agricultural waste, example wood shavings, sawdust, corn cobs, etc., and even human and animal waste. See, when you are going to produce a single cell protein and you are going to market it, it needs to be profitable. So, it means that whichever is the microbe which you are growing as a protein supplement, media composition or the substrate on which this organism grows has to be easily available, and at the same time it would be environmentally friendly if we are going to utilize some biomass which otherwise goes as a waste. So, normally we use inexpensive substrates like agricultural waste, wood shavings or even human and animal waste. In addition to direct use as single cell protein, microbes also contribute to protein demand when they are used to upgrade the protein content or the quality of fermented foods. So, one is use the microbe directly as a protein supplement, as a single cell protein. Other aspect is, in an already prepared and marketed foodstuff, you can add up the single cell protein so that the altogether protein component of the marketed product gets increased and it will become more bioavailable. Although microbial protein provides a relatively small proportion of current human nutrition, the growing global demand for protein is likely to make single cell protein increasingly important. High growth rates or ability to utilize unique substrates like carbon dioxide or methane result in processes which offer much higher efficiency and or sustainability than is possible from traditional agriculture. So, what we are looking at here is, your traditional agriculture will provide certain amount of foodstuff. In addition, if you can give supplement which can be produced as in a higher rate, utilize waste or use components like carbon dioxide or methane which otherwise would not be utilized, then this would be more sustainable and overall the efficiency of food production will also increase. Research on single cell protein technology started a century ago when Dilbruck and his colleagues found out that the high value of surplus brewers yeast as a feeding supplement for animals. During World War I and during World War II, yeast single cell protein was employed on a large scale in Germany to counteract food shortages during the war. So, the utilization of single cell protein to combat the shortage of food started about a century ago during the World War I and II itself. The food from oil idea became quite popular by 1970s when Chaminade being awarded the UNESCO Science Prize in 1976, and paraffin fed yeast facilities were being built in a number of countries, and the primary use of the product versus a poultry and cattle field. The Soviets were particularly enthusiastic about this technology, and they opened large BVK protein vitamin concentrate plants next to their oil refineries in Kostovo and Kirishi. So, use the paraffin waste and the petroleum waste to get it converted into protein vitamin concentrate, which can be used as a cattle feed. The Soviet Ministry of Microbiological Industry had 8 plants of this kind by 1989. However, due to the concerns of the toxicity of the alkanes in single cell protein and pressured by the environmentalist movements, the government decided to close them down or convert to some other microbiological processes. Corn. Corn is a range of vegetarian and vegan meat substitutes made from Fusarium venenatum mycoprotein, which is sold in Europe and North America. Algae, fungi, especially filamentous fungi and yeast, and bacteria, all these can be used as a single cell protein. And in the future, dietary proteins may also be derived from proteins which are secreted by engineered microbial cells. For example, your milk or egg white proteins and produced from animal and plant cell cultures in which the cells are no longer microbes, but they are either not animal cells nor plant cells either. Thus, what is SCP or single cell protein and what is the other types of protein which are used as a supplement, the line becomes faintly blurred because we can use multitude of sources as the technology advances. The microorganisms utilize the carbon and nitrogen which is present in these materials and convert them into high quality proteins which can be used as a supplement in both human as well as the animal feed. The single cell proteins can be readily used as a fodder for achieving fattening of calves, pigs, in breeding of fish, etc. And this also offers an unconventional but a plausible solution to the problem of protein deficiency being faced by the entire humanity. The conditions, one is nutritional. They are high energy value, protein content has to be high, amino acid balance has to be considered. The second is the technological conditions. We require to know the type of culture and the nutritional requirements of the organism which is going to be used to produce the protein supplement. The third is the toxicological aspect. So, when you are going to say I am going to produce a single cell protein, one is who is the target, that is human or animal. Then what is the nutritional value of the product which you are targeting to synthesize, what is the protein composition and what is the amino acid balance. That is regarding the consumer. The second is the technology which you are going to use. So, depending upon the culture or the microbe which you are going to use to produce the protein, what is the type of culture which you are going to go, then what type of conditions do you require, the medium or the substrate, the pH, the conditions, what are the special features are required for it to produce the type of culture, the protein product which you want. 3. During the process of the production of the protein which you are going to extract and utilize, are there any toxicological aspects or any other byproducts which can be cause either allergenicity or toxicity to the humans which consume. So, these are the 3 conditions which need to be standardized or taken into account before you start producing a single cell protein. Among the fungi, Aspergillus fumigatus, Aspergillus niger and Rhizopus cyclopeum are the ones which are commonly used. Among the yeasts, we have the Saccharomyces cerevisiae, Candida tropicalis and Candida utilis which are being used. And among the algae, the well known ones are the Spirulina, Chlorella pyrenodosa and Chondrus crispus. Bacteria used are Pseudomonas fluorescens, Lactobacillus and Bacillus megaterium. Single cell protein is currently produced from a limited number of microbial species, especially we consider the humans as the major consumers. The production steps for single cell protein generally includes 1. The preparation of nutrient media, possibly from waste, because then the waste can be efficiently utilized. This becomes more environment friendly. 2. The cultivation, including solid state fermentation. 3. Separation and concentration of the single cell protein, in some cases drying it. And final processing of the single cell protein into ingredients and products. The production of pure microbial cultures for a desired protein product requires a nitrogen source, the source of a carbohydrate, and other nutrients like phosphorus to support the optimal growth of a culture. So, any standardization, so like growing any microbe, we need to look into a carbon source, a nitrogen source, then other nutrients like phosphorus, the organic or inorganic components, the pH, the temperature. So, all those conditions have to be standardized first. 2. Contamination has to be prevented, because you are going to grow a specific culture for the production of a specific protein. The components of the culture media are either heat sterilized or filtered through a microporous membrane. Then you inoculate. So, once the media has been prepared and sterilized, you inoculate the selected microorganism into it. Most of the processes are highly aerobic except algal fermentation. Hence, a good supply of oxygen is indispensable for the production of a single cell protein. After the multiplication of the biomass, it is recovered from the medium and purified further for enhanced usefulness or for storability. Single cell protein from algae. Microalgae, which are produced for human or animal consumption, typically have a very high protein content, normally 60 to 70%. They also provide fats, the omega-3 fatty acids and carotenoids being of particular interest, vitamins A, B, C and E, minerals, salts and chlorophyll. Microalgae are currently used in the form of supplements available as tablets, capsules or liquid form, but they are increasingly processed as ingredients which can be used in pastas, in baked goods, snacks, etc., etc. The most accessible commercial products are derived primarily from Arthospira platensis and Arthospira maxima. This is sold as spirulina, marketed by Earthrise Nutritionals. Then you have chlorella. This is marketed by Taiwan Chlorella Manufacturing Company with this trade name and trademark. You have the marketing Duaniella salina, again by a company. This is primarily for as a source of beta carotene. Then Avenazonum Flows aquae. This is marketed by Blue Green Foods, the Klamath Valley Botanicals, again as a source of supplement. Algae generally feed on carbon dioxide and light, although some products like the algavea are produced by traditional fermentation rather than by photosynthesis. So, algae as a source of single cell protein, we can either have photosynthetic products or you can have fermentation products. Single cell protein from fungi. Fungi, which are grown as single cell protein, will generally contain 30 to 50% of the protein. The amino acid composition compares favorably with the FAO guidelines. Threonine and lysine content is typically high in a fungal protein, but methionine content is relatively low, but it will still meet the FAO or the World Health Organization's recommendations. The methionine content of some fungal products like Marmite is even lower, and also sulfur containing amino acids have been enriched in single cell protein from Chromomyces fragilis by cultivation on whey. So, if you think that a particular product is lacking or contains a lesser percentage of a particular amino acid, if you change the substrate or the media conditions, sometimes you might be able to enhance the amount of amino acid, the particular amino acid which you are looking for in the product concerned. Low density lipoprotein cholesterol has been reduced in volunteers who consume the mycoprotein from Fusarium melanatum, and blood glucose and insulin levels may also be favorably affected. So, when the study was done with a trial population, one half being given fed with a single cell protein, mycoprotein, and the other half being the control which was not given. They found that the LDL cholesterol was reduced in those people who were fed with the mycoprotein. The blood glucose level as well as the insulin levels also were in a favorable condition when compared to the control population who were not fed on the mycoprotein. So, this actually demonstrated that mycoprotein can not only act as a supplement, but also becomes a healthy supplement. The Cone brand was launched in 1985 by Marlow Foods in UK, and the Cone products contain mycoprotein from the filamentous fungus Fusarium venenatum. The fungal biomass provides a texture that resembles the meat products, and this normally is maybe the only SCP product which is exclusively used for human nutrition and has been extensively branded. Spent brewer's yeast, that is Saccharomyces cerevisiae, have been sold for more than a century in yeast extracts like Marmite, which is marketed by Unilever and Sanitarium Health Food, the Vegemite by the Biga Cheese Limited, Synovis by Glistagenic AG, and Vitam. These yeast extracts provide a good source of 5 important group B vitamins, but also of proteins. Another commercially available yeast, Torula. This is from Candida utilis, which has been renamed as Pichia jardini. It is a widely used flavoring agent, and it is also a very high source of protein. Torula was used in Provenstine, which is produced by Provista Corporation in the 1980s, along with similar products using Pichia and Clubberomyces yeast. Torula is rich in the amino acid glutamic acid, and for this reason it has been used to replace the flavor enhancer monosodium glutamate MSG. A process called Specilo was developed in Finland to produce single cell protein for animal feed from the sugars which are present in the sulfite liquor of a paper mill effluent. The filamentous fungus Pachyomylis variety was grown on sugars including pentosis in the sulfite waste liquor or on the wood hydrolysates. There were 2 factories operating on Finland in Manta and Jamskoi during 1982 to the 1991 period. But as the cellulose mills stopped their activities, these factories were also closed up. Although the product was sold as an animal feed, it was also investigated as a supplement in meat production like sausages and meatballs. British Petroleum pioneered production of yarrow EA for animal feed from waxy and paraffins from an oil refinery in the 1970s, building a pilot plant with up to 100 kiloton annual production facility. The product itself was considered safe, but the plant failed to get the required production permits because of the environmental concerns. Bacterial single cell protein usually contains 50 to 80% of the protein on dry weight basis, and essential amino acid content is expected to be comparable or higher than the FAO recommendations. Imperial Chemical Industries developed a single cell protein for animal feed from methanol using the bacterium Etylophilus. This protein contains 70% of protein and it was used as a pig feed. Protein, however, could not compete with the cheaper animal proteins or animal feeds which were available at the end of 1970s, and finally the production was discontinued. Calista Inc., another company, opened a production facility for the product feed kind in UK in 2016, and it is partnering with Cargill to build a larger production facility in the USA. So, this is a feed kind product mainly used as an animal feed. Let us see what are the advantages of using single cell protein. Microorganisms have a very high rate of multiplication. Hence, large quantity of biomass can be produced within a short time. These microbes can also be genetically modified to vary the amino acid composition. So once you genetically modify them, you can ask them to produce a single cell protein with the particular amino acid which you want, or those amino acids which you want to be present in a higher rate than what is present in the natural state. Broad variety of raw materials, including waste materials, can be used as a substrate. This will also help in decreasing the number of pollutants, so environment friendly. The production is independent of climatic conditions. What are the disadvantages then? High level of nucleic acid in the biomass makes it difficult for consumption, as it may lead to gastrointestinal problems. The biomass may trigger an allergic reaction if the digestive system recognizes it as a foreign product. The presence of the nucleic acids in high content also leads to elevated levels of uric acid. In certain cases, the development of kidney stone and gout have been seen if it is consumed in very high quantity. Possibility of the presence of toxic metabolites which result in hypersensitivity and other skin reactions is also probable. The capital cost of production is very high, as sophisticated machinery is required for the production of the single cell protein. Now let us see the applications. It is a source of an instant energy. It is extremely good for healthy eyes and skin. It provides the best protein supplemented food for undernourished or malnourished children. It serves as a good source of vitamins, amino acids, minerals, crude fibers, etc. It is used in therapeutic and natural medicines for controlling obesity, reducing blood sugar level in diabetic patients, reducing body weight, cholesterol and stress, prevents accumulation of cholesterol in the body. It is used in the cosmetic products for maintaining a healthy hair, for the production of herbal beauty products like biolipsticks, herbal face cream, etc. Used in the poultry industry, as it serves as an excellent and convenient source of proteins and other nutrients. It is also used for feeding cattle, birds, fishes, etc. So, in a nutshell, if you can have a technologically sustainable methodology for the production of single cell protein, you can culture it, whether your source is a bacteria, fungus or an algae. It has to be sustainable. The cost of production as well as maintenance has to be profitable. Then you have a very cheap source of single cell protein, which can not only be used as a protein supplement, but it can also be used in multiple phases, for example, cattle feed, animal feed, in cosmetic area, as a therapeutic product, in so many different fields. But you have to make it sustainable. At the same time, work has to be done or research has to be done also to find out which are the other microbes or the other organisms which can be used for single cell production, single cell protein production. So, the questions which we face are, what about the other organisms which can be used for recipe production? And the second are, what are the regulatory issues which have to be concerned with? See, whenever we talk about a food product, there are, since it is meant for either human or animal consumption, we have to talk about allergenicity, we have to talk about toxicity. Even if the product per se is not toxic or allergenic, it can produce metabolic byproducts which in turn can produce these results. So, research or work has to be done to find out whether any allergenicity or toxicity is involved. Then regulatory issues like the standard FAO and the WHO regulations, which are required not only for the process, for the technology, but also for the product certification. All these have to be taken into account before we can commercialize any particular single cell protein concerned. Thank you.